Welcome to another Explainer. We've got an incredibly fascinating journey ahead of us today. Whether you're a food scientist, a healthcare professional, or really just someone who loves diving into how things work, you're going to want to hear this. We are looking at a class of biological catalysts that are literally reshaping modern industry. I'm talking about lipase enzymes. So let's jump right in and uncover the sheer biological potential of these powerhouses across the industrial landscape.
To give you a roadmap for this explainer, we're going from the micro scale right up to global industry. We'll cover 1. Introduction to lipase enzymes. 2. Structure and catalytic mechanism. 3. Sources and classification. 4. Enzyme production and purification. 5. Industrial applications. And finally, 6. Immobilization and future perspectives.
Let's kick things off with Section 1, Introduction to Lipase Enzymes.
Okay, before we get bogged down in the biochemistry, I want you to consider the sheer scale of what we're talking about here. Back in 2021, the global enzymes market was valued at an estimated $11.4 billion US. And get this, it's projected to hit a massive $17.88 billion by 2028. Why the explosive growth? Well, it all comes down to what enzymes actually do. They're proteins that speed up biochemical reactions without being consumed or altered themselves, a vital process called catalysis. Industries everywhere are waking up to the fact that enzymes are a highly efficient, pure alternative to the harsh chemicals we've traditionally relied on to get things done. Now, the history of lipases is essentially a timeline of continuous scientific breakthroughs. It all started way back in 1856 when Claude Bernard discovered them in pancreatic juice. For decades, animal pancreatic extracts were the absolute go-to commercial source, but things really kicked into high gear in the 1990s as industrial demand spiked. By 1993, scientists mapped the very first 3D structure of a bacterial lipase, specifically from a microbe called Pseudomonas glumi. And then, just a year later, a total game changer, Novo Nordisk introduced lipase, which was the very first recombinant lipase to hit the market. Moving right along to Section 2, Structure and Catalytic Mechanism. Once scientists started actually mapping out these 3D structures, they noticed something pretty incredible. Even when lipases from entirely different species showed absolutely no sequence similarity, they still shared a common folding pattern. It's known as the alpha/beta hydrolase fold. Picture a central, mostly parallel, 8-stranded beta sheet, and it's sandwiched on both sides by alpha helices. This highly specific structural blueprint is the core foundation for their amazing catalytic abilities. So how does this structure actually do its job? Well, the catalytic mechanism of lipases during ester hydrolysis happens in 2 highly precise steps. Think of it like a biochemical dance. Step 1. There's a nucleophilic attack by a serine residue right on the carboxyl carbon of the ester bond. This forms a temporary covalent acyl enzyme intermediate, which kicks out an alcohol, specifically diacylglycerol. Then comes step 2, hydrolysis. Water swoops in, launching a second nucleophilic attack that breaks that intermediate and finally forms a carboxylic acid. It's incredibly elegant and precise. All right, Section 3, Sources and Classification.
How do we organize all these different lipases? The modern standard is the Lipase Engineering Database, which essentially sorts them into 3 main classes based on their oxy and ion hole structure: GX, GGGX, and Y. But practically speaking, where are we actually finding these enzymes out in the wild? The short answer is everywhere. They're naturally found in plants, animals, and microbes. Plants pack them heavily into their seeds to turn fats into sugars during germination, and animal lipases, as we know, help digest dietary fats. But for heavy-duty industrial work, microbes, like bacteria, fungi, and yeast, are the absolute favorites. They give us massive yields, broad substrate specificity, and honestly, they're just much easier to genetically manipulate.
Fungi, in particular, are considered some of the absolute best sources out there, mainly because they secrete their lipases directly into the environment, making extraction a whole lot simpler. Interestingly, though, bacterial lipases usually perform optimally in alkaline or basic environments. Fungal lipases, on the other hand, strongly prefer acidic conditions. And that key difference right there is exactly why scientists hunt for so many different microbial strains. It gives industries an incredibly versatile toolkit of enzymes that can handle wildly different chemical environments. Which brings us to Section 4, Enzyme Production and Purification. To meet global demand, we rely heavily on 2 main scale-up methods. First, there's solid-state fermentation, or SSF. This uses a solid substrate with very low moisture to anchor the cells. It's a method traditionally favored in Asian countries. It boasts really high volumetric productivity and produces less waste effluent. But removing metabolic heat is tough, making large-scale operations pretty difficult. Then you have submerged fermentation, or SMF, which submerges the microbes right into an aqueous solution. Western countries really developed this in the 1940s to mass-produce antibiotics. While it does take longer to cultivate, SMF allows for easy agitation, precise parameter monitoring, and highly standardized scale-up using massive bioreactors. But of course, once the microbes produce the enzyme, you can't just ship it off right away. It has to be purified. For those extracellular microbial lipases, we start by salting out the proteins using ammonium sulfate precipitation. Next up is dialysis to pull out the salts. And finally, the real precision work happens using advanced column chromatography techniques. Things like ion exchange chromatography, using DEA Cephros, and gel filtration are used to isolate the pure lipase based entirely on its electrical charge and molecular weight.
Let's look at Section 5, Industrial Applications.
Once purified, the real-world impact of microbial lipases is just staggering. Take the food industry. They use lipases to modify lipids, completely transforming the texture and sensory properties of our food. In the pharmaceutical world, they target molecules with immense affinity and specificity, helping create advanced digestion therapies and synthesizing very specific drug compounds. But it really doesn't stop there. They remove lipids in cosmetics, clean animal hides in the leather industry, break down oil in wastewater treatment, and they're even a major player in producing biofuels and fine chemicals. Because they can happily operate in organic solvent media and microaqueous conditions, they are basically indispensable across this entire wheel of global industries.
Finally, Section 6, Immobilization and Future Perspectives.
Now, to be completely fair, we do need to acknowledge some realistic hurdles here. While processing based on lipases is incredibly promising, rolling it out everywhere is sometimes slowed down by two main things. First, the comparatively high cost of the purified enzymes themselves. And second, when you just use free-floating enzymes, they sometimes lack the extreme catalytic stability needed for some of the harsher, continuous industrial applications. But scientists have a brilliant workaround for this: enzyme immobilization. Instead of just letting the enzymes float freely in a solution, researchers actually anchor them to solid supports. They do this using techniques like physical adsorption, covalent bonding, entrapment, or microencapsulation. Why go through all the trouble? Because locking them down drastically boosts their thermal and ionic stability. Plus, and this is a huge deal for the bottom line, it lets the industry physically retain the enzyme and recycle it from multiple reaction batches. That significantly slashes those high costs we just talked about.
Which leaves us with a final thought to chew on today. Could optimizing these incredibly resilient enzymes through advanced immobilization be the ultimate key to revolutionizing green industrial chemistry? When you think about it, we've gone from a simple digestive protein found in a pancreas back in 1856 to a multi-billion-dollar catalyst driving the sustainable production of everything from biofuels to pharmaceuticals. The biological potential of lipases is truly remarkable. Thank you so much for joining me on this explainer, and keep exploring the science that shapes our world.